First of all, Araceae is the family of monocotyledonous floral. The main objective of this study is to determine the distribution of Araceae in UiTM Pahang Forest Reserve Jengkar Pahang. The another objective is to examine the Araceae distribution and light intensity, soil pH, moisture and organic matter. The results show that the soil moisture for location 3 are more higher than location 1 and 2, while the light intensity in riverside more higher compared to land region. Members of this family have the widest range of physical traits like form, habitat preference, developmental pattern, phenology, and adaptive modification for effective pollination. Approximately 105 genera and 3,000 species make up the large family of Araceae. They exclusively found in the tropics for 90% of the genera and 95% of the species, showing the predominantly tropical distribution. Besides that, Araceae is highly varied and offers a range of purposes, including those for food, decorative plants, and medicinal. Aside from that, malaria is one of the several Araceae plants that may be used to treat both the illness and its symptoms. For example, in Togo, entire plants are not particularly used to treat malaria, but they are used to alleviate some of the disease symptoms such as fevers and headache. The problem statement from this study is: first, Araceae were common in mature forests, but they were uncommon in secondary forests and plantation. Second problem is natural resources have not been handled successfully, as there have been since the general population is unaware of the several possible applications for Araceae tubers. And last but not least, the majority of the Araceae species fail to flourish under harsh conditions. There are three objectives from this study. Firstly, to determine the distribution of the Araceae in UiTM Pahang Forest Reserve Jengkar Pahang. The second one is to examine the Araceae distribution and light intensity, soil pH, soil moisture and organic matter. Lastly, to identify leaf area of family Araceae using ImageJ software. Significance of this study is to investigate the richness and distributional trend throughout an elevation at ecological significant places. Because of elevational fluctuation, regional differences in temperature and soil types, the presence of hilly terrain lead to the development of multiple diverse ecosystems in a comparably limited area. The gathered information will also help scientists evaluate the success of ecological communities that were purposefully devoid of Araceae species when they were employed for ecological studies. Furthermore, will demonstrate the existence of many aroids with distinct geographic distribution. Members of the Araceae family, commonly known as aroid, have a very varied range of ecological characteristics, including epiphytic, climbing and terrestrial plants, as well as aquatic that are submerged, emergent or free-floating. Besides that, Araceae family quite diverse and offers a variety of uses. They are mostly ornamental plants. In Amazon area, the most important species of the genus Philodendron are used to treat malaria and other associated fevers. Their leaf, roots, petals, fruits, and rhizome of plants are used to manufacture therapeutic medicines. Ecological factors such as soil also impact the distribution of Araceae species. Soil pH has a significant impact on plant growth. The optimal pH range for soil is about 5 to 6.5, although soil with pH value that are too low, 4.5, or too high, above 7, can impede or even stop growth and development. If plant is growing in soil that is not appropriate for that species, it will be stressed and its capacity to produce will be impaired. The leaves of Araceae species are often arranged spirally or only very rarely. In contrast, the petal is frequently well developed and carries a shade to safeguard the young. Some aroid, which is genus Anthurium and Monstera, have a geniculum that enables them to orient the leaf blade in respect to the light in the most advantageous manner. The leaves of Philodendron meridionale are broad to extremely wide, oval in shape, simple and petiolate. Aroid flowers are often borne on the fleshy axis known as the spadix, are frequently many and small and are seldom reduced to a small number. The fruit of the Araceae family include raphidian berries. The seeds might have ribs or not. The big testa is made up primarily of the parenchymatous cell with the raphid sacs, occasionally pigmented cell, and rarely sclerotic elements. The method for morphological characteristics of RSA species involve the sampling method, image acquisition, and leaf area measurement using image processing techniques. For apparatus and materials that used in this study were different selected species of the RSA, which is Alocasia morphontanensis, Monstera deliciosa, Philodendron giganteum, Philodendron hederaceum, and Syngonium podophyllum, while the apparatus were a ruler, pencil, white A4 paper, measuring tape, double tape, and smartphone. The first method is sampling method. The leaf of 5 RSA species were specifically chosen, were collected, and were legitimate in terms of their physical trait. Both quantitative and qualitative observation of the leaf morphological properties were made. A ruler was used to choose and measure the leaf of varying size. Additionally, the color, form, arrangement, texture, border, and venation of the leaf were studied and observed. The second method is image acquisition. A line of 5 cm was drawn on a piece of paper by using a pencil and a ruler for calibration purposes in automatic measurement for image processing. All 3 leaves of the 5 selected species were photographed using smartphone camera. Each leaf was positioned in parallel to the calibration line during the photography. The photo then saved into the computer and uploaded into ImageJ software for the image pre-processing steps. The next step was to submit the photos of all leaves from the 5 species into the program. Using the rectangle tool, any problematic region on the photograph will be cropped off, including any clip edge, shadow, or other imperfection. The photo then improved by minimizing the look of shadows and modify the amount of light. The photo then converted to 8-bit and the threshold was changed in order to measure the necessary parameter. Finally, ROI Manager was utilized to choose the species leaf area. For the method, ecological factors, which is soil and light intensity, to the distribution of RSA species. For the raw materials, RSA plants, pH meter kit, jars, distilled water, electronic balance, and lux meter. Firstly, the sampling method. For the samplings, no plots will be constructed and random plot were selected. The step to measure the soil pH is first, 3 soil from different regions were taken and labeled as 1, 2, 3. Then 20 grams of the oven dried sample for each plot, measure and place it in a flat bottom test tube. Water then added to each sample soil, then 50 ml of distilled water added to the test tube for each sample. The test tube is closed with the stopper and mix the sample with the water by shaking the solution. The samples then left overnight. After that, the electrode will insert into each sample in the tube and the data for each soil sample were recorded. The step to measure the soil moisture is the 20 grams of soil for 3 plots, weighed on the balance and weight recorded. Then place it into the crucible and weigh again on the same balance. After that, place the soil into the oven to let it dry at 100 Celsius for 1 hour. The step will be repeated until there's no weight changes for each samples. To measure the soil organic content by using the formula C2 minus C3 divided by C2 minus C1 and times by 100. C2 is the weight of the dry and clean crucible. C2 is the weight of soil samples with the crucible, and C3 is the weight of the soil samples after cooling down by desiccator for 1 hour. The step to measure light intensity by using lux meter. The sensor cap on the lux meter tool will be removed. The sensor of the lux meter will be placed face down and roughly 1 meter from the ground at the sample sites. The sensor will then be turned on, all of the buttons will be depressed, and the instrument panel's number display will be read and noted. Lastly, data analysis. The resemblance of each habitat was discovered using the Principal Component Analysis (PCA) of the PAHANG software. Habitat with a high degree of resemblance will develop in the same cluster. Abiotic data such as tomography, light intensity, soil temperature, air temperature, air humidity, soil moisture, soil pH, and soil organic matter are utilized to create clusters. Alocasia morphontanensis is evergreen, simple, dark green leaves are alternately arranged. This species is petiolate. The petiole is about 38 cm to 73.2 cm. They have angulate margin, reticulate, venation, and are deltoid and petiolate. Alocasia morphontanensis also produce a solitary white spatha. The leaf length about 30 cm to 49 cm, while the width is about 11.5 cm to 41.5 cm. The second genus is genus Monstera. From this study, the species that I found under this genus is Monstera deliciosa. Monstera deliciosa has the potential to be converted into antibacterial medication due to secondary metabolite substances such as tannins, steroid, and alkaloid. Monstera is leafy plants with a thick stem that has a diameter of 2 to 4 inches and capable of growing on the ground or up a tree. The petiole length for this species is from 90 cm to 20 cm, while the width is 23 cm, where the leaf length is about 27.5 cm to 29 cm. The third genus is genus Philodendron. From this study, there are two species under this genus, which is Philodendron giganteum and Philodendron heteroecium. For the leaf of Philodendron giganteum have a heart-shaped leaf that are entire and unpinnate. Due to the calcium oxalate crystal present, this species is toxic in all in its part. Furthermore, the petiole length is about 35.5 cm to 42.5 cm, while the width is from 11.7 cm to 14 cm. The second species for genus Philodendron is Philodendron heteroecium. The leaves are alternating, simple, cordate, acuminate, and glossy. The leaf blade is longer than the narrow petioles. The petiole length is about 4 cm to 7 cm. The width is 4.7 cm. Large, free, and lanceolate are present. Initially bronze in color, leaf rapidly become green and envelope stems. The leaf length is about 7.5 cm. The flowers are white and tiny. Last but not least, genus Syngonium. From this study, the species under this genus is Syngonium podophyllum. Their leaves are arrow-shaped, tapering leaf tips, grooved leaf base, and pinnate leaf spine. Along the stem, the leaves are arranged alternately. When the leaves are broken, a white sap comes out. The leaf length usually about 22.8 cm, while the width is about 8.8 cm. The fruit is enclosed in a red spit and develop the form of a brown sin cap. According to table 5.4, Alocasia morphontanensis, Monstera deliciosa, Philodendron giganteum, Philodendron heteroecium, and Syngonium podophyllum score 1 for leaf color, which means they have a green leaf. Next, Philodendron giganteum and Philodendron heteroecium score 4 for leaf shape as their leaves are cordate, while Alocasia morphontanensis score 3 as this species have sagittate. Leaf shape for Monstera deliciosa score 2 as it has a pinnatific. Last but not least, Syngonium podophyllum score 1 as this species has an elliptic leaf. For the ecological factors, which is soil pH, table 4.5 show the result of soil pH for each different location. The results show that location 2 has the best soil pH for the Araceae species, which is 5.02. This is because most of the Araceae species prefer the pH range between 5 to 6.5. If the pH soil too low or too high, it might hinder or stop the growth and development. Table 4.6 show the result for organic matter for each sample soil. Based on the result, the location that has highest organic matter is location 1 and 2, which is 4%. Due to a wide range of microbiological activities, location 1 and 2 has the larger level of organic matter than the location 3. The organic matter in soil is crucial for sustaining life. Organic matter also supplies the surrounding plants their primary nutrients. Table 4.7 show the moisture of each sample soil. Araceae species might get root if soil have too much water in it. The germination, growth, and overall development of Araceae plants depends on sufficient soil moisture. A lack of water can result in stunted development, drying out, and even death. Location 1 has the best soil moisture compared to location 2 and 3. This is because there's a lot of changes in soil weight, means location 1 has higher moisture level. As the primary energy source for photosynthesis, light intensity is crucial for plant development. Every plant needs a certain amount of light to develop. The riverside region has the maximum light intensity, which is 2076. Riverside places typically have more light intensity than land areas, particularly along the ocean, where there is little to no interference from large buildings or dense vegetation. The light intensity in land lower than riverside region. This is due to denser vegetation like that found in forests and woods can form a canopy in land location, reducing the quantity of sunlight that reaches the ground. The Araceae family morphological research has made it possible to classify the species according to their order, genus, and species. Both quantitative and qualitative qualities were examined, measured, and documented in order to completely comprehend the morphological trait and correctly categorize the species. In conclusion, all of the chosen species have been able to be identified correctly after considering all of these qualities. Although species from different genera might have the same morphological characteristic, it is typical for those species from the same genus to share more similar traits than those from different genus. Additionally, this research was able to successfully utilize the use of image processing technology to scan and measure the leaf areas because of the many sorts of distinctive characteristics that the leaf show. As for suggestions for further investigation, more thorough investigations are required for fully understanding the morphological characteristics for numerous species in Araceae family. This is done in order to find and maximize the advantages that may be derived from this family, and to make sure that future generations have access to knowledge about their species. Besides that, the Araceae district data must be updated periodically to keep up with the evolution and adaptability of this species to the changing environment. As a result, ongoing research on the species of the Araceae family is essential and required. Other than that, exploring more about the economic and cultural significance of Araceae. For example, in economic aspects, the species of Philodendron, Alocasia, and Spathiphyllum are few well-known examples due to their attractive and distinctive leaf. These plants are frequently utilized in botanical gardens and as houseplants, while in cultural aspects, certain Araceae plants are utilized in rituals.